My thesis is titled Impact of Burn Frequency on Arthropod Communities in North Carolina Longleaf Pine Savannas. My name is Paige Turner, presenting on behalf of the Biology Department, mentored by Dr. Sally Kerner. My study takes place in the North Carolina Longleaf Pine Savanna, which is a fire-dependent ecosystem. Fire is required to limit the intrusion of invasive hardwoods and woody shrubs that would otherwise dominate the understory. Deforestation, agriculture, habitat fragmentation, and fire suppression all threaten the 900 species of plants that don't exist anywhere else in the world. Arthropod diversity is essential for the health of the savanna because they perform many roles. Herbivores have a positive influence on above-ground plant biomass. Detritivores break down litter and debris between burn periods, and many compose the bulk of the diet for other inhabitants of the savanna. Fire suppression and burn frequency limit biodiversity by altering competition dynamics. Plant species composition, canopy density, and root competition, which are impacted by burn frequency, can change the diversity of arthropods as many have evolved alongside select species and will leave the savanna in search of the conditions they prefer. There are few publications on the arthropod species composition of the longleaf pine savanna. 40% of species are declining at a rate of 2.5% every year. 70% of plant species in the longleaf pine understory require insects to reproduce, so without them, the conservation of this ecosystem may not be possible. The purpose of this study is to determine the impact of disturbance regimes on arthropod communities in the North Carolina longleaf pine savanna. My hypotheses are that the site with the most recent burn and no disturbance from logging will have higher arthropod richness overall, that the site with disturbance will have higher species richness than those without, and that there will not be a significant difference in order diversity and evenness between disturbed and undisturbed sites. A transect sweep net was used to collect larger arthropods, such as plant bugs and crickets, which are associated with above-ground plant biomass. A modified leaf blower was used to vacuum a majority of common microinvertebrates, and hand collection was used to pick up larger macroinvertebrates that are not targeted by other methods used. This is a schematic of the site layout where you can see that 4 square meter plots were randomly selected within 2 25 square meter blocks of each 1,000 square meter site. The sweep net was performed separately from the 2 blocks in an area not disturbed by my previous collections. The collections took place in July and August during the most active hours for insects in the savanna. The collections were timed for consistency, and samples were stored in a cooler in the field before being moved to a freezer in the lab. I also surveyed plant species composition with Alyssa Young and used that data to compare to my arthropod community data. Of the 1,028 arthropods collected, 984 could be classified to family and 118 were classified to species. On the right is what a sample looks like after all insects have been separated from the debris. I found the highest abundance of arthropods in Site 2, which had a disturbance from logging and was burned every 2 years. The bottom table shows the total number of plant species recorded at each site. There was no statistically significant difference in the number of plant species at each site. Site 3 had the highest order diversity. The table at the bottom indicates that there was not a statistically significant difference in order diversity between either of the 3 sites. Site 3 also had the highest family diversity, with 79 total families identified. The table on the left shows the total number of individuals in each order at each site. 132 individuals composed 14 orders in Site 1, 475 composed 15 orders in Site 2, and 421 composed 19 orders in Site 3. The order composed of the most individuals was Hemiptera, with 276 total collected. The highest peak in the graph of order richness is the 163 Hymenoptera collected from Site 2. It's important to note the orders which are absent. Coleoptera and Thysanoptera, which are beetles and soil-dwelling thrips, were absent from Site 1. The table at the bottom depicts the Simpson's diversity index for order evenness at each site. A higher Simpson's index for Site 2 signifies some orders are more abundant than others, which was illustrated by the peak in Hymenoptera. In conclusion, my study supported that flying invertebrates are the first to return to burned blocks of the savanna, as indicated by the higher prevalence of Hemiptera and Orthoptera in Site 1, and the abundance of ground and soil-dwelling arthropods such as Coleoptera and Thysanoptera. I found that it takes more than 8 weeks for the arthropod community to recover from prescribed burn. Site 2 abundance was elevated in comparison to the other sites, which was likely a result of being a refuge for arthropods seeking to escape the burn. The Simpson's index showed that there was evenness across arthropod orders and no indication that one was outcompeting others to disrupt community dynamics. I also found that shelterwood cutting, a regime restricted to Site 2 in this study, enabled species richness. Therefore, all 3 of my hypotheses were supported by my results, to the extent that they could be, considering a majority of the collected arthropods could not be classified to species. For future studies, I would suggest expanding the range of collections and using a wider variety of methods to capture a more comprehensive view of the arthropod community. I would like to thank Dr. Sally Kerner, Alyssa, Will, Catherine, and other Kerner Lab members for their assistance and support during this project, and a special thanks to Dr. Omar Ali for awarding me the Dean's Undergraduate Research Award, which funded my fieldwork and allowed me to devote so much resources to this study. Thank you for watching.